So I want to start by sharing with you a real conversation I had yesterday with my one of my supervisors, Professor Joan Brugge. When I told her I'm planning to go on the job market this year and hopefully interview. So she asked me, "Oh, so are you going to fly to Israel?" And I said, "I don't know." And then she said, "Ah, so you're going to interview in person?" And I said, "I don't know." And then she said, "Ah, so you're doing hybrid?" And then I said, "Maybe." And then she said, Yaara, because that's how they call me in the US, she said, "Check the policy." And I was like, "What policy?" And I think this kind of conversation reflects a lot of the uncertainty we have currently in our life, where we're trying to make plans, but we really can't because we don't know if it will come back. But I want to say that we're actually super, super privileged because having the sense that normality will endure is something that many cancer patients do not have, where for them, the question is not, will it come back, but rather when. So despite the many years of cancer therapy, of developing new targets and new cancer therapies, it's still very challenging to cure cancers. And too often do we see that patients that had an amazing response to first-line therapy come back, and sometimes months, years, sometimes even decade after an initial successful response. So why does cancer come back? And what has recently emerged as one of the reasons is this concept of cancer persister cells. So what are persister cells? Persister cells are a subpopulation, a rare subpopulation of transiently drug-tolerant cells that, while they themselves are resistant, they give rise to sensitive progeny consistent with a non-mutational mechanism. Now, the cancer persister cell field is very young. Cancer persister cells were first described only in 2010. But it's not a young field in general. Persister cells, or cells that can survive therapy without having any underlying genetic mutations, were first described in microbes more than 80 years ago. So when I was wrapping up my PhD, I studied microbial evolution, and I saw this emerging field in cancer. I said, this would be a really great field to switch to. So, we packed our family and we moved to Boston. And then it occurred to me that, well, cancer cells are not exactly like bacteria. And one of the really biggest differences is that in microbial persistence, we know that it's tightly linked to dormancy. So bacterial cells can survive therapy because they're not cycling. But this doesn't seem to be the case in cancer, where some persisters can cycle. So I'm just going to show you a picture of what happens if you take non-small cell lung cancer and you treat it repeatedly with a very high concentration of the same drug that is given today in the clinic. Then yes, it kills most cells, but you see that after 2 weeks, you have these rare cycling persister cells. These are less than 0.5% of the total population. And as far as we and many others have looked at, there are no mutations associated with this phenotype. So why can some rare lineages cycle? And to try to address that, we developed the watermelon vector, named after its green and red fluorophores, and also because I'm a proud Israeli and I love watermelons. And this is not a fruit, it's a lentiviral construct that allows to combine proliferation tracking with lineage tracing. So the proliferation tracking comes from the red component. The red component is an inducible H2BM cherry. So when we add dots, all the cells become red. And then when we chase dots away, as the cells divide, the color is diluted. And this now gives us a very nice handle. Here is a colony of cells after 2 weeks of drug treatment. And despite the fact that they're rare, we could use flow to sort them out. Now, the green component allows us to do lineage tracing, where we infect cells such that each mother cell would have a unique barcode. And because this barcode is integrated into the cells and expressed, when we do single cell sequencing, all the daughter cells that belong to the same clone would have the same lineage barcode. So we brought this system to try to address how persisters cycle in cancer. So we took a non-small cell lung cancer cell line, we generated a watermelon library, and we generated this watermelon PC9 line. And what we did is we took like this 2 weeks time course where we'd add 3 dots at the beginning. So most cells die, only some cells can become persisters. And during this time course, we sampled cells for single cell sequencing at day 0, 3, 7, and day 14. And altogether, we were able to harvest 57 cells from 10,000 lineages. So how does that data look like? The first thing we did is we put this into an algorithm and we said, please organize the cells according to similarity and transcriptome. Now, despite the fact that the algorithm was oblivious of the time course, it ordered the cells according to time. Day 0, 3, 7, and at day 14, you could see that the non-cycling cells are following a complete distinct trajectory from the cycling cells that look somewhat, some of them look somewhat like the untreated cells. Now, the beautiful of this system is you can take each of these cells and extract from it the lineage dynamics. So here, each line is the lineage, and this is time. And you can appreciate that many lineages disappear, but some lineages do not only disappear, but also undergo a massive expansion throughout the time course. So again, we combine this data together and ask, what is special about the lineages that are able to proliferate in the presence of drug? And what we found is that these lineages, these cells upregulate progresses associated with ROS. So here is on the x-axis time, and 2 of these genes that are associated with cycling persisters. And I just want you to notice that this expression is associated specifically with cycling under drug and is not expressed, is not associated with cycling per se, because drug naive cells, zero cells cycle very rapidly, but they don't induce these genes. This also holds when you go to the program level. So here each dot is a cell, and we look at the NRF2 signature, which is the main antioxidant regulator of the cells. And again, we see an increase in the cycling, moderate in the moderate, and much lower in the non-cycling. And don't trust single cell unless you see a phenotype assay associated with it. So indeed, we went in and we measured if we see a ROS, if we can see ROS, directly probing for ROS, and we see a very strong time-dependent increase in ROS. So now we had a model where most cells, when you add drug, die. Some cells persist, but they are arrested because they have high ROS, meaning that if you could alleviate ROS, then you could enable more cells to cycle. So to try to validate that, we alleviated ROS both chemically and genetically. And in both cases, we can see that by doing that, we could increase the fraction of cycling cells. Now, I want to say that the beautiful thing about building a system, as we heard in our last talk, is that once you have it going, you could generalize it and do it also in other models. So we started with one model and one cell line, and we expanded it to 8 different cancer models, and we treated them and derived persisters with the same drug that it used today in the clinic. And indeed, we see that in many of these models, the cycling cells are associated with a transcriptional adaptation as well as an accompanying metabolic adaptation. So I know some of you are saying cell lines are nice, but what about humans? And the beautiful thing about working with a field that is very, very young is that there are a lot of groups around the world who are trying to help and generate data. And while we were working on that, came out a study from UCSF that analyzed persister cells that were derived from real human patients with the same drug that we were using in our models. And what we saw there, that also in humans, there is an increase in the ROS signature. But what was even more interesting is when you stratify the patients according to if their cells are cycling or not, and you could somewhat do it computationally, we see that this difference in signature exists only when you look at cells at persistence phase and is lost once genetic mutation is established, suggesting that there are these signatures that enable cells to survive before genetic mutation kicks in. So when we started this study, we're very much focusing about this regrowth phase, but the truth is we know very little about cancer persister cells. We don't know why some patients relapse earlier than others, and really have very limited understanding and why and how some of these persister cells can be the reservoir from which mutation, resistance mutation emerges. So to sum up my talk, if there is one thing I want you to remember, is that we really know very little about cancer persister cells. And that means we should be very optimistic, because maybe within this world lies a target or a way to really, really cure cancer and give these patients the long-lasting normality that they all deserve. So I want to thank the many people who contribute to this work. It's been a team effort across multiple institutions, and I'll be very happy to take questions.
Thank you very much, Yaara, for this great talk. I see already that Slil, one of our speakers, have a question. And next, Dan Mishmal. Yeah.
Yeah, that was such a beautiful talk. Thank you so, so much. So I wanted to, maybe I missed this and you already mentioned it, but I'm assuming that the mechanism of persistence is going to be very intimately related to the mechanism of the drug, right? And so if you go back with something that's completely unassociated with the first drug, that remind me again what it does. I think it has something to do with proteasome inhibition.
So this is actually an EGFR inhibitor in this case.
An EGFR inhibitor. Sorry, exactly. So if you go back and try it with a second drug, do you find similar links to ROS, or is it a completely different persister mechanism?
And so it's an excellent point. The mechanism of persistence is very much linked to the mechanism of resistance and what the drug directly inhibits. So we have other models where we use, for instance, chemotherapies, and we see different mechanisms associated with the adaptations of persisters. So it's really what the cell is trying to cope with, right? And indeed, we expect it to be specific both to the target, what the drug is targeting, and to the lineage, because different cells have different ways to cope with stress.
Thank you, Professor Dan Mishmal. You have a question?
Yes, I do. So the first thing that came to my mind when you spoke about when you mentioned ROS is, of course, my favorite organelle, the mitochondrion. And I wonder, when you mentioned that the persistent cells do not accumulate any kind of mutations, how did you check that? Did you check that in whole genome sequencing, only exome sequencing, only RNA what did you do to make sure to look for mutations?
So I want to say first that persister cells, and I haven't shown that, including the cycling cells, once you withdraw the drug, and expose their progeny to drug, they're sensitive. Okay, so the mutational mechanism, which was done very heavily around the system that we're using, they don't have any mutation. And also the drug that we're using, they cannot develop a mutation in vitro. You need like really, really long, you need a very patient postdoc. But I think going to your second question about mitochondria, this is indeed something that I am very interested in, because we do see that there is association, this transcriptional change is associated with switch to mitochondrial energy production and fatty acid oxidation, right? So one of the things would be maybe there is some priming in the mitochondria that enables them to be persisters. And that's one of the things that I plan to follow up when I start my own lab. And we have no beautiful ways to combine mitochondrial lineage tracing and metabolic assay together with all this world, I would say, of non-genetic resistance. Yeah.
This is
so stay tuned.
Well, I will, and I would offer you also to go to study not only the mitochondria but the co-regulation between the nucleus and the mitochondria. But you know, we can talk offline.
We'll talk.
Yeah. Thank